For our last section of Chapter 10, we're going to be talking about protein synthesis. This is another way to say how we make proteins. DNA gives us the ability to make proteins, and that's probably one of the most important functions of DNA, is helping us to make proteins. Because proteins, we talked about them a lot this year, they help us carry out reactions, because a lot of proteins act as enzymes. They help us move things around, they help us grow. So proteins are really, really important to make, and DNA helps us do that. As we talk about how proteins are made, we're going to be talking about a couple different molecules. First, we'll be talking about DNA, obviously, and we'll talk about how we take some messages from DNA and we turn it into mRNA, which is a little bit different type of genetic molecule. And then we'll take that mRNA and we will use it to get some building blocks for proteins. And those building blocks for proteins, if you remember from this year, are called amino acids. So when we look at the flow of information here, we're going to be moving from our DNA molecule to a slightly different molecule called messenger RNA, mRNA. And then that messenger RNA is going to help us get some building blocks and actually make proteins. So that's the general overview of what we'll be talking about on the next couple slides. So on this diagram, this gives us a good visual of what we're going to be talking about on the next couple slides. We're going to be starting with our DNA molecule, that twisted ladder that's inside of the nucleus. Then what we're going to do is we're going to do a process called transcription. A transcript, this means to write. So basically, we're going to write a message, make a message from that DNA. And we're going to make that message and turn that message into mRNA. So that's still in the nucleus. And then, after that step takes place, we're going to move that message out of the nucleus to the cytoplasm. And then we're going to do one more process, and it's going to be called translation. We're going to translate that message into the actual protein, into the actual molecule. So there's going to be some other steps we have to talk about along the way, but that is the big overview of what's going to be happening as we make proteins in our cells. So just some basic differences between RNA and DNA. We said that DNA is a double-stranded, it's a twisted ladder. If you look at our picture, RNA is actually going to be a single strand, so it's only like half of the ladder. DNA has a different sugar in it. The sugar in DNA we talked about is deoxyribose. The sugar that's in RNA is going to just be called ribose. It's a little bit different. It has like an extra oxygen in that sugar. One more thing that's a big difference between these, we talked about our 4 letters for DNA being A, G, T, and C. For RNA, we do not have a T. We do not have a thymine. Instead, we have uracil. So we have a U instead of a T. So for RNA, the 4 letters are going to be A, U, G, and C. They're going to be a little bit different. In DNA, it was A, T, G, and C. So the big idea, RNA has a U and DNA has a T. That's going to be the difference in their letters. There are 3 types of RNA in our cells. The first type is messenger RNA, and its job is going to be to take a message from our DNA and use it later on to make a protein. Another type is called transfer RNA, and sort of looks like an upside down T. Oftentimes it's called tRNA for the transfer part, and the fact that it looks sort of like a T helps me remember that. This type is going to actually bring the building blocks of the proteins together to help make the protein. Okay, so those are 2 different types. And then ribosomal RNA, that's going to be the genetic material that helps make up the ribosomes. Those are the little cell parts. They're pictured in that middle part of our picture. Those are the little cell parts that help make proteins. when we did different diagrams with them with our cells this year, you guys made them as little circles, little dots. That's just a zoomed in picture of one over towards the left. So once again, here's a good review of what each of those types of RNA do. The messenger RNA carries the instructions from the genes in our DNA to make a protein. So it acts as a messenger. It carries the message from DNA in the nucleus to the ribosomes in the cytoplasm. Ribosomal RNA or rRNA, this is going to be where protein synthesis takes place, where we make proteins. This happens in the ribosome. So the ribosome is the cell part where proteins are going to be made. Our third type, our transfer RNA, is really important because this helps transfer or bring amino acids to the ribosome to help make that protein. So it sort of carries the building blocks that's going to help make our protein. So those are your three types of RNA and what they do. So when we're making a protein, the first thing we want to do is going to be to make a message from our DNA molecule. We want to make that mRNA. In order to do that, we're going to have an enzyme called RNA polymerase, and it's going to bind or connect to a certain part on the DNA called a promoter, and that's going to cause the two DNA strands in that twisted ladder to unwind and separate. When that happens, we're going to have complementary or matching RNA nucleotides that are going to be added and joined by that enzyme called RNA polymerase. So just like we talked about how we did DNA replication, we made a copy of the side of the DNA ladder. We're doing a similar thing here, but instead of making a DNA copy of one side of the ladder, we're going to make an RNA copy. And that is going to be just a little bit different because the copy that we make is not going to contain the letter T. It's going to contain the letter U where there would have been a T because the RNA does not have the letter T in its code. So it's going to make that copy, and it's going to keep on making that copy until it reaches a termination signal. So an area on the DNA that says, hey, this is where the message needs to stop. It's sort of like a period or an exclamation point saying, hey, this is where we stop. And then that's going to be where the RNA polymerase releases from the DNA and also releases that new RNA message. So this entire process is called transcription. We have transcribed or written a message from our DNA, and we've written that message in RNA code, an mRNA molecule. So here is a picture of what this looks like. Our first step over towards the left, we have our DNA molecule that's being untwisted by RNA polymerase. So we can see that that ladder has been separated, and we have two sides of the ladder now that are not connected where that RNA polymerase is. Then that RNA polymerase is going to start to make a copy of one of the sides of those ladders, and it does it by matching up A and U and G and C this time. So where there's an A on the DNA, the mRNA copy is going to match it up and put a U there, because in RNA, A and U go together and G and C go together. Remember, there's not going to be a T. There's no thymine that's being made in that RNA message. So we copy that side of the ladder and we make this little RNA message. Once the RNA polymerase reaches a termination signal over towards the last picture, that's going to tell that enzyme to stop making the copy. So it will detach from the DNA. It will also let that message go away from the DNA, that RNA message, and we can see that the DNA goes back to being that twisted ladder. So basically what we do is we untwist that DNA ladder for a little bit, we make a copy of some of that information there to make a protein, We let that DNA ladder go back, and then now we have this new RNA message, this single strand message that we are going to take and use to make a protein. So we have transcribed a message, and we're going to use that message to make a protein. Before we do that, just a little review of what proteins are made of and what they look like. Remember, proteins are made out of amino acids. Those are the building blocks, the monomers of a protein. And those amino acids are held together by peptide bonds. There are about 20 different amino acids that we use to make proteins, so we have a huge number of proteins that we can actually make because we have so many different building blocks. The order in which those amino acids are joined determines the structure and the function of proteins. Over towards the left, we can see that proteins can take many forms, many sizes and shapes. So proteins can end up being very, very large, complicated molecules. And a lot of them are pretty big and they are pretty complicated. This helps them do a lot of things in our cells and in our body. So mRNA codes for the specific amino acids that are going to build the protein. In our message, we're going to look at every 3 nucleotides or every 3 letters in our message. For example, AUG. Those would be 3 letters that would code for an amino acid. Each 3 letters or each 3 nucleotide groups is called a codon. So every 3 letters in our message is called a codon. Each codon, every 3 letters in mRNA, it codes for one specific amino acid, or it could code for a start or a stop signal. So we're going to either be coding for a certain building block to come in and add to this protein, or we'll code to start the protein, or we will code to stop the protein. Those are things that our codons are going to do. This follows the rules of our genetic code, and these are the rules that relate to how a particular sequence or order of nitrogenous bases corresponds to a particular amino acid. So this genetic code basically tells us, okay, if you're given these 3 letters in your mRNA, what amino acid does that give you? And this genetic code holds true for all of the organisms on Earth. We all share this genetic code. So that's something that is really special about our DNA. We share a lot in common with all of the other organisms in their DNA as well. So here's a picture to help explain this a little bit better for you as well. In our picture here, we have a mRNA message that we have made from the DNA. We can see that it's twisted a little bit, but it's single-stranded. It's not a 2-sided ladder. It's only a 1-sided ladder. It's not double-stranded. So if we look at our message and see what letters are contained in it, we have an example over towards the right of the letters that could be contained. We can see that there's, let's see, 1, 2, 3, 4, 5, 6, 7 times 3. There's 21 letters in this DNA, or sorry, this RNA molecule. So every 3 of those letters is considered a codon. So our first 3 letters up towards the top are G, C, U. That's a codon, and that's going to tell us either to start a protein or to bring a new building block into that protein. So we can see in this example there are 7 codons. So there are 7 groups of 3 letters. So that's what we're looking at when we see a codon. It's a group of 3 letters that basically make up a word in our genetic code. And that word is going to be an amino acid. So this complicated looking chart shows us the overview of our genetic code. This shows us that the 3 letters that make up or give us each amino acid. So for example, the 3 letters U, U, U. If you were given that codon, that would give you the amino acid called phenylalanine. That would be one of the building blocks in that protein. If you were given the 3 letters U, U, C, that would give you the same amino acid, phenylalanine. And we can see that we have codes for all of these amino acids. Some of them have like 4 possible codes. Some of them maybe have 3 or just have 1. We can also see on here that there is a start code, there is a stop code. So this chart lays all that out for you. If we were in class, we would get more in detail with this. You guys would be practicing this a lot, but I do think this is a little bit complicated because it's so new. It's kind of hard to do this in the virtual class setting. I think it's just best right now if we just look at the general overview of it. Don't worry about the things on the side that say first, second, or third. For those bases, that would be a way that you would figure out what 3 letters would give you. But on the middle of that chart, you have the codes for all of those amino acids that we're going to be using. So that's the idea here. We can see there's a whole bunch of codes, and there's 20 some amino acids, as well as some stop and start signals that will exist in our genetic code. So in translation, we're going to take the RNA message that we transcribed during transcription, and we're going to translate it or turn it into a protein. And we're going to talk about just a couple steps of translation to see how this takes place. The first part is called initiation, and this is going to be where a tRNA molecule and the mRNA molecule join together. So the codon that we were talking about, remember, that's on the mRNA molecule. That's a 3-letter group on the mRNA molecule. It's going to match up with something called the anticodon on the tRNA. The tRNA has the amino acid attached to it. So in our picture down below, we can see that we have the mRNA strand, this very, very long strand in this picture, and then we have a ribosome, this round ribosome that's attached to it. There's actually 2 of them. Inside of that ribosome, you can see it like an upside down T, the tRNA. In that picture, the tRNA is matching up to a certain part, a certain codon on that mRNA, and when it does that, it's going to start to build a protein. We can see on that tRNA molecule, it has a round amino acid on it. Over towards the picture on the right, we can see that we're starting to build a protein. So we have that blue circle that's making it up. Now it's also being attached to a red circle, a red amino acid. So we're starting to build a protein from that message. That part is called elongation, where we make the protein longer. So we keep on adding amino acids to it to make, at the end, a chain of amino acids. And that will be how we start making our proteins. So this elongation process will continue as the ribosome moves a distance of 1 codon on the mRNA. So for every 3 letters, we're going to be adding another amino acid on. We can see in the picture down below our amino acid chain, all those different colored circles getting longer and longer. Eventually, though, the ribosome will reach a stop codon. It'll hit a stop codon signal on the mRNA. That part is called termination, and that's going to be where the building of the protein ends, and we then go into disassembly. That's going to be where the ribosome goes away from that message, and then that new protein that we made, also sometimes called a polypeptide, is released. So now we have this new protein molecule that we made from that, and that is basically the end of translation. We've translated our mRNA message into a protein. So in summary, there are three big processes we've talked about with DNA. The first big one was replication, and that was where we made a copy of the DNA from DNA. So we've taken a DNA molecule, we untwisted it, and we made copies of those DNA molecules, and that gave us a new DNA molecule that had half of it being old from the original DNA molecule, and then half of it being new, being a part that was built from new nucleotides. Transcription was making RNA from DNA. We transcribe a message, so we untwisted the DNA and we make an mRNA message. That's transcription. Translation is where we take that message and we use it to make proteins. We use the ribosomes to do that. We use the transfer RNA to do that. So that's going to be where the actual proteins are made. So those are three big processes that you want to make sure you don't confuse. So make sure you go back in your notes and review each of those to see how they're different and see what's happening in each one of those. One last thing about our DNA. Sometimes you'll hear the term human genome being used. This refers to the complete genetic content of humans. So when we look at the DNA of humans, we see that there's about 3.2 billion base pairs in the 23 human chromosomes. So a lot of letters we're dealing with, a lot of rungs of that ladder. So there's a ton of information there. And it took us years to figure out what all of those letters mean. Some of those letters we still don't know what they mean. We don't know what they code for. Some of them don't code for anything. So there's still some information we're trying to figure out about our human genome, about the DNA that makes us up. Knowing about the human genome allows us to know which gene sequences control particular biological functions that may help diagnose, treat, or prevent genetic disorders, cancers, and other diseases. So knowing a lot about our DNA tells us what gene is coded for by which part of the DNA. We can look for when things go wrong. We can look to see if something's happening with that gene. Sometimes we can do different types of gene therapies. We can look at what medications might be helpful for that. So this plays a huge role when understanding how we treat different illnesses and diseases, and it tells us a lot about how humans get all of their genes and how we have changed over time. So DNA is going to be one of the things that we continue to learn more about as we go through our lives. Scientists are working on this stuff all the time.